Welcome to Microdose Psychedelic Insights, powered by the Conscious Fund. This is the PsyFi series, discovering the cutting-edge science and research in psychedelic medicine.
Welcome back, everybody, to another exciting episode of the PsyFi podcast, where we talk to leading industry experts, clinicians, and researchers to unravel the mystery that is psychedelic science. I'm very excited for today's episode. I have the fortune of recording in sunny Mexico, and I'm here with Dr. Marvin Hausman of Pilz Biosciences. He's conducting some really exciting research, and I'm really grateful that you're joining us. So thank you so much for coming on the show, Dr. Hausman.
Well, I'm glad to be here. Thank you for inviting me. Oh, you're in sunny Mexico. I'm in sunny Florida, so that's okay.
Yeah, that's good. We're both in a warm place.
Trying to move to a position where we don't have much glare in my glasses. I think that's a little bit better. So we'll see.
Yeah, you look great. So, Dr. Hausman, it'd be wonderful. You know, we've had a lot of opportunity to connect, and you had great reason at the Mushroom Conference in November. But for our listeners, if you could just introduce yourself and tell us a little bit about you and your work, that would be a fantastic way we could start this conversation.
Well, I'm a medical doctor. I graduated from New York University School of Medicine. I also was an immunologist during medical school, and I helped do some of the early research on the HLA system, which types people for kidney transplants at that time. And my career then went on to general surgery, and then I was drafted temporarily into the Marine Corps for Vietnam, and my orders were changed, and I was sent to NIH, where I did further immunology research on the complement system, and I was one of the people who uncovered the third and fifth component of complement with Dr. Ralph Snyderman at NIH. He was my mentor. And then I was accepted to the transplant program at UCLA, and I became a kidney transplant surgeon, and then went on to work on dialysis, and my career took off. I was a drug developer for Bristol-Myers and for Squibb. I developed some well-known drugs. Then I formed a company that went on the New York Stock Exchange called Medco Research, and we then had our own cardiovascular drugs. In 1998, the company was purchased by King Pharmaceuticals. I went on into Alzheimer's disease, formed a company called Axonyx. I was one of the founders, was co-founder, and we then merged that company into Torrey Pines Pharmaceutical. And then I became very involved in nutrition. I turned my attention to mushrooms, and not the psilocybin type. This was just nutritional mushrooms.
Sure.
There are about 10 major edible mushrooms, and I started looking for substances in mushrooms that are produced inherent to the nature of the product, but that can't be reproduced in humans. So there are substances in nature that we need, and I set out to prove that we don't manufacture. One of them is something called ergothymine, which is an amino acid in mushrooms. And that amino acid cannot be produced by humans or mammals. But you have a transport system, and that was discovered by the Germans, by Dr. Grundemann. And about 10 years ago, Dr. Grundemann showed that you had in your stem cells, white cells, and red cells, that there's a substance, an ergothymine transporter, a protein in the cell membrane. And the only function of that transporter protein is to move ergothymine into the human cell. Now, why would you have a transporter and you can't manufacture the passenger for the transporter? How important is this substance in life? And the Germans went on to hypothesize that you couldn't have human life without ergothymine, that ergothymine neutralizes singlet oxygen, S-I-N-G-L-E-T, singlet oxygen, which is oxygen that's unbalanced, it's -1. And so when life moved from the sea onto the land, and I try not to get involved in religion or politics, I'm just thinking of evolution.
Sure.
Cells started producing oxygen and killed themselves because they were producing singlet oxygen. When they figured out that they had histamine in their, histidine amino acid in their cytoplasm, they said, oh, we can manufacture ergothioneine. And you can only manufacture ergothioneine naturally from histidine. And so they then were able to neutralize the single lead oxygen, and life began. And I've been fascinated with this concept of ergothioneine. Then vitamin D2 is also produced by mushrooms. Humans produce D3. It's not a vitamin, it's a hormone, by the way. It's misnamed. A vitamin is something that humans can't produce. If you produce it, it's not a vitamin. But in those days, they thought that it was a vitamin. So D3 is really a hormone, a prohormone that becomes stored as a hormone, and then it's released when you need it, and it becomes activated. Mushrooms produce D2, which is similar to D3, and we now feel that D2 supplements D3. So by eating mushrooms or taking D2 supplements from mushrooms or ergothioneine, you're supplementing major components of your general nutrition system. And then I was approached about, in June this year by a group in Germany that says, Hausman, with your background, maybe you'd like to help a company in Canada, which was called Pilz Bioscience Corp. And they said it's psilocybin. I said, well, psilocybin is natural. It comes from mushrooms. I said, I love it. So, and that started my relationship with Pilz Bioscience. And then I chose to show a need in a devastating disease. I chose autism because it's unmet. And whether in your own family or other families, you always come across a family that has an autistic relative or child. It's a disease that's unmet in terms of diagnosis and therapy. And so that's what I've been focused on.
Wow, that is an incredible answer, Dr. Hausman. There's so many different facets of that introduction I want to comment on, and I'd love to share and talk about. I think your work with HLA is so exciting. As we talked about, my first author paper is in the third time lung transplantation or second time lung retransplantation, and looking at how that correlates with death risk. And it's just been really fascinating to be able to take my clinical experience and scientific background and bring it into the psychedelic space, and to be able to understand people like you and the work you've done and the contributions you're making. It's all very exciting. So I guess my next question would be, now that you are finding yourself doing research in this very exciting and dynamic space, what are you most excited about? And it's really interesting. You talked about how mushrooms produce these compounds, or we have substrates, or we have pathways in our body that require us to be able, that only work with compounds in mushrooms. So we talked a little bit about biomarkers that you're looking at. These are all things I'd be interested to hear more about. But just first, it'd be interesting to know, what you're looking forward to now that you're working in the psychedelic medicine space.
Well, do we have 5 hours? I mean,
I know we don't have 5 hours.
This is quite a question. The first thing that excites me is that it's an open pathway. Mm-hmm. The psychedelic research is a child that's just being brought home from the hospital. The mother had just delivered the child. We're just beginning psychedelic research. We have to have it catch up to medical technology. Medicine is way ahead of the psychedelic industry. Psychedelic industry is based on subjective behavioral changes, and I feel that we need to use hard, objective evidence in medical technology to prove out the psychedelic field. I mean, isn't it exciting to come into a field where you can add something? And for example, I think autism has subsets of the disease. Why do they call it a spectrum? Spectrum means it's a lot.
Right.
There are probably subsets. So when you don't know what it is, you call it a spectrum. Autism spectrum disorder. There may be 10 subspecies of autistic children or subsets of disorders. And so I find it exciting to grab onto that and to run, so to speak, with that football, because it gives me creativity every day to think of what can I do today that's different from yesterday. And that's what that keeps me alive.
Absolutely. And so I would love to know a little bit, like you talked about the field right now, it being so nascent. A lot of the markers that people are looking at are subjective. Is your depression better? Do you still qualify for having PTSD? But it's so fascinating that you're looking at an issue like autism and that you're looking for concrete biomarkers. So how are you going about doing that? What are some biomarkers you're looking for right now in your research? And, what do you hope to find?
Well, let's take example of let's simplify it down. You're a patient and you're not feeling well. You go to the doctor and he says, "Oh, you have high blood pressure, and I'm going to put you on this pill to lower your blood pressure." So you go home. You come back to the doctor a week later, and he says, "How are you feeling?" You say, "Oh, I feel great. You're a genius."
Yeah.
And then he said, he says, "I don't, I feel wonderful. You were wonderful." Well, the doctor says, "Don't you think we ought to take your blood pressure to see if it went down?"
Yeah.
I mean, I understand the
Right.
subjective feeling that you're feeling better. And that's the same thing in the field of medicine when you use a bias, a subjective behavioral analysis. They're very important. Behavioral analysis is very important. So the whole beginning of the psychedelic field is obviously based on behavioral changes and moving people into a different space mentally. But for example, if everybody says, and I'll give you another example, psilocybin works through serotonin. It's a serotonin response.
Right.
Well, 95% of serotonin is produced outside the brain. Only 5% is produced in the brain. Why is that?
Yeah,
Why
the rest is produced in our
the
gut.
rest of the body? Why did the process of development put, or whatever force you believe in, put serotonin in the periphery? And by the way, the serotonin that's produced in the small intestine, for example, cannot cross into the blood-brain barrier because it's hydrophilic. It attracts water. And you have to have an active mechanism to move it into the brain. So it doesn't move in. So that's a conundrum. And that's what I want to prove. I want to see, is it involved in diabetes? Is it involved in liver disease? Where
Wow.
besides autism is it involved? Why did nature put it in the mushroom, which is a food that acts on serotonin, and 95% of the serotonin is produced in the small intestine and the bone marrow? We've got a lot
Wow.
to learn here. And that brings us to the microbiome. So
Right.
a human being is composed of 30 trillion human cells and 100 trillion bacteria. We're 3-to-1 bacteria. Bacteria, if you think about it, control us. We're controlled by bacteria that makes up our body. We don't even know that. So are bacteria controlling our mind? Are bacteria controlling serotonin release? And that's what I'm looking at diagnostically, to look at bacteria and the changes genetically in bacteria. In autism, for example, I'm taking one model, being very specific, and also looking at cytokines, these inflammatory molecules that are produced by bacteria and by other cells in your body. So it's a three-legged stool. It has three legs. One leg is behavior modification. The other leg is the microbiome. And the third leg are inflammatory molecules called cytokines. And if you take any one leg off, you're going to fall down, aren't you? The chair is not going to support you. So that's my approach to this disease called autism, and it's my approach to the psychedelic industry.
I love that. I really love how you're looking at the microbiome, and I have noticed some of the research that's come out lately that's correlated the impact of the microbiome to autism spectrum disorder. So I think that's a really valuable and novel pursuit that you're taking with your research. One of the things we talked about that a message you wanted to get across to people was how to actually properly design and conduct these preclinical and clinical trials in the psychedelic space. And I know you know a lot about the proper way to really design an effective trial. So maybe you could share a little bit about that and how you're going about designing the trials you're conducting now.
Well, my clinical expertise has always been in designing clinical trials. I've been designed or helped on 6 to 7 NDAs, which are new drug applications with the United States government
Wow.
for big companies, including my own. So there's an art form to doing that. You have to look at life like a chess game. You move your pieces. Medical research is no different than playing chess. You move your pieces forward. You don't want to move them back. You don't want to make a mistake because people suffer when you make mistakes. And one of the things that you do before you start research is to look at the literature. Look at what's out there. Like, for example, when I was looking at autism, I found that there was 1 or 2 articles that showed that when you treat autistic children with antibiotics, they had improvement in their symptoms. Why?
Wow.
And that pointed me towards the microbiome. And then we find that when there's stressful situations, that autistic children, for example, deteriorate in terms of their behavior. Why? Why does stress do that? Is it inflammation? And I had worked on post-traumatic stress disorder and suicide, and we think that suicide may be an inflammatory reaction. That's why people commit suicide.
Wow.
And
That's a really exciting, it's a really interesting, sorry to interrupt you. It's a really interesting angle.
And I did this work with Michael J. Fox Foundation. We did a Parkinson's study.
Wow.
In an animal. We took the human gene, it was done by another company, and put it into the brain of the mouse. And the mouse then has not autistic, but Parkinson-like behavior.
Right.
But if you look back in the history of Parkinson's disease, Dr. Parkinson studied, so they called it after his name. Most of those patients had intestinal problems. They had irritable bowel syndrome. And when we looked at the mouse model, that mouse had intestinal issues as well. So the theory now is that this neuroinflammatory condition called Parkinson's disease doesn't start in the brain. It starts in the small intestine with bacteria. And these bacteria produce cytokines that migrate up the vagus nerve into the substantia nigra, a particular area of the brain where Parkinson's disease occurs. So
and then does it kill off dopamine neurons in the brain at that stage?
Well, you know that L-DOPA, we don't know that, but first of all, dopamine, again, is a molecule that's more produced in the periphery than the brain. It doesn't cross the blood-brain barrier, but you have to give something called levodopa, Levodopa, which is a precursor to dopa, and that crosses the blood-brain barrier, and that creates dopamine in the brain. So we do know, and so you treat Parkinson's patients with levodopa.
Right.
But the concept I want to get is the interaction between the microbiome and disease processes. And again, inflammation as well. Parkinson's is a neuroinflammatory disorder. Alzheimer's is a neuroinflammatory disorder. Depression could be a neuroinflammatory disorder. Depression is, if you're a manic depressive, that's a genetic disorder. But acute depression is probably a neuroinflammatory process. And what role?
I wonder if, a conversation that's been happening a lot in the psychedelic space lately is that there must be some central mechanism or something that is in common to all these different disorders, be it depression, PTSD, anxiety, end-of-life anxiety, for example. There must be something core and central to them all that, we're finding psychedelics are able to work across the board for all these different conditions so effectively. And after talking to you, it seems that, a lot of people are thinking maybe there's some foundational or basic brain mechanism that it's acting on, but there's a possibility that it might be altering our gut microbiome, and that could be the central focus of all of these different conditions, and that's what you're researching. I think that's so interesting.
Well, Gaurav, you're talking to me, right? So there's a language between us, English language. We're now using English. But there's a language in the body. These bacteria are talking to our cells. I don't mean wax like I'm on hallucinogenic drugs and talking to you right now, but I'm telling you, there's a language we don't know. And when you say the role of the microbiome, well, they're producing molecules, language that communicates with receptors and organs in the body. We're now looking at the brain, but what receptors is psilocybin communicating in the liver? What receptors is it communicating in the small intestine? Can psilocybin be a treatment for irritable bowel syndrome or Crohn's disease or celiac disease? Why is it produced by the intestine? I think the jury is still out, and that's what excites me. I know we have companies that are mainly looking in the brain and looking at use of psilocybin in cancer and depression, looking at psilocybin and other. They say you're opening up another space in the brain where this client or patient can go to, and it's healing. But what is the mechanism? What's the underlying mechanism of action of these hallucinogenic compounds, these tryptamine derivatives? And that's what I'm setting up in autism, using that as my focus, and to look at the intestine, to look at fecal changes in autistic individuals, to look at inflammatory markers or the language of inflammation in animals with autism, in humans with autism, and also look at behavioral modification. So we have an ongoing study now in Italy with a VPA rat, a valproic rat. Valproate is an anti-epileptic drug. So if you have a pregnant mother, this is where they, I think, uncovered the idea here, and the relationship is you treat this pregnant mother with valproate for epilepsy, she has an increased tendency to have autistic children. So Dr. Treza in Italy has created a model, and other science have validated this model, where you treat the pregnant rats with VPA, valproic acid. And the pups, or the children of these rats, are autistic. They have behavioral abnormalities. They have social interactions that are abnormal. And that's what we're studying. But on top of that, I've overlaid a paradigm of microbiome analysis, of cytokine analysis. And so we've already laid the groundwork. So we started this study in July this year. And we have a lab in Vienna, Austria, where we're measuring cytokines with a company called Thermo Fisher, which is one of the largest genetic companies in the world. And in Canada, we're measuring microbiome changes genetically with a company called Microbiome Insights. So now we have discovered biomarkers specific to autism that can be potentially modified as you see your behavioral changes. So they were control animals and severely behavioral modified animals because of valproic acid. And we now have that groundwork. And now we're going to treat a similar population with psilocybin. And then we're going to compare the changes in behavior with the modulation of the cytokines and the microbiome. So that study is underway, and we hope to start the treatment program the beginning of January.
Wow.
We will have some data coming out next year.
Well, that is so fascinating, the studies that you just talked about going on. It's really cool that you're correlating changes in the microbiome in the gut to these valproic rats that have been administered VPA, so their pups have show autistic characteristics. So I guess one thing I can't help but wonder about, let's say that you do because research pans out the way you want to, and it turns out psilocybin is able to attenuate autistic symptoms or effectively treat the disorder. Something I talked with Jackie about, which is really interesting at the mushroom conference, is what would this translate to as far as an actual therapy goes for autistic children in the future? Would that entail giving them psilocybin? Of course, that raises some really interesting ethical concerns. Do you think maybe it would be some sort of non-hallucinogenic derivative or analog of psilocybin, or what do you see this looking like down the road when it's actually going to make it to humans and autistic kids?
Yeah, no, it's an excellent question. First of all, what we're going to establish, hopefully in the first quarter of next year, 2021, is what is the dosing that's going to get a response in the VPA treated, in the VPA population of rats. Then we can translate that dosage to a human dosage. And then the next step would be pharmacokinetic studies in humans. Obviously, we're not going to do children right away. We're going to do adults. But we're also going to be looking for data in the microbiome changes and the cytokine changes, the inflammatory markers. So we're now thinking ahead. As I said, life is a chess game. Research is a chess game. And there's something that you use in the cannabis world called entourage effect. You say you have an entourage effect because the whole is more effective than the individual components. So nature put other derivatives in the weed, the cannabinoid. Cannabinoids. You have THC, you have CBD1, CBD2. It goes on and on and on, different receptors, and people talk about an entourage effect. Well, I want to show whether there is an entourage effect in the psychedelic field. So I am now producing other derivatives. You'll hear about it. We're going to probably make an announcement in the near term that we are going to be one of the first companies to create an entourage formulation for autism.
Wow.
Formulation, we're picking certain derivatives that may lower the side effects, which you just said, of psilocybin. Then we're also going to be looking not just in big doses, we're going to be looking at micro dosing. And with the derivatives, we may need small doses that are very safe in children. And we hope to have a lot of this knowledge next year. I think 2021 is going to be quite a year for the research we're doing at Pilz Bioscience Corporation.
Oh, I think so too. I'm really, really excited to see the exciting developments and the new steps in the research that you guys are taking. And a lot of things came to mind. I guess one of the questions I have is, do you think that you'll be able to, in one or two treatments or in a protocol of treatments, actually cure autism, or do you think through a microdose formulation or some an entourage effect that's specifically formulated for autism, it will just be able to way more effectively attenuate the side or the symptoms of the disorder, but maybe not actually cure it at its core?
I have to quote a guy who I admire. I never met him. His name is Plato, okay?
Okay.
Philosopher. And Plato said, you never know the answer, you only approach the answer. So
Good answer.
I put my pants on one leg at a time.
Yeah.
There's no way I can predict the answer. But on the other hand, if I think outside the box, I mean, you just gave me an idea. If I modulate the microbiome, what if I combine psychedelics with antibiotics?
Yeah.
We may
There's
not.
a lot of options there.
I haven't even thought about it till right now as we're talking. I mean, maybe the psychedelic has to be in combination with an antibiotic regimen, and maybe we could reverse this condition. But I think we have to differentiate a genetic cause of autism versus a developmental cause of autism.
That's a really good differentiation you made, a developmental versus genetic cause. Because another thing I was thinking about, like in these rats, you give them VPA, and that the litter will be born with autistic symptom. So maybe, just since we're speculating in this podcast, 'cause that's what we do, there's a possibility to identify that our progeny is going to be autistic before they're born, and possibly give them beneficial factors or drug treatments when they're still in the womb to maximize their chances of maybe not being autistic when they're born, or having the best likelihood of success.
So my daughter is, I have several doctors in my family. My son is a
Okay.
regional anesthesiologist. I told them none of them to go into medicine. They don't listen to me. So my daughter is a maternal fetal medicine doctor. She's the smart one in
Wow.
the family. She's brilliant.
Uh-huh.
Probably everybody says their daughter's brilliant, but she is. She graduated one of the top medical schools and the top of her class in the United States, Yale, for example.
Wow.
She graduated from Yale, one of the top medical schools.
Wow. That's
So
impressive.
about a year ago, I was researching the microbiome. I was researching ergothymine. Uh, let me just turn off this phone here. Get started. Sorry about that.
No, it's all good.
I should have turned it off before. So I said,
No
Nicole,
worries.
I just came across this article where when the children who are born by C-section have a deficient immune system,
Oh, wow.
and compared to those who are born through the vaginal canal. So I called up my daughter. Maternal fetal medicine she does high risk pregnancy with
Right.
mothers and children in the utero. So I said, Nicole, are you aware of this? She said, Dad, we knew this about 2 years ago. And what we do when we have a C-section child, we take a cotton tip swab and we transfer the microbiome from the vagina into the nose of the newborn child.
Wow. Whoa. I never
Think about
considered I never considered that would be the reason why, that they're missing out on some of the bacteria through a vaginal birth that they're not getting through a C-section. And so then they take a swab and they actually introduce the
Yes,
bacteria through the
they do
nasal canal. That's
right after
mind-blowing.
birth, right after delivery from the C-section. But think about that. We're just touching on it now with some of these diseases.
Yeah.
I mean, can we modulate the development, the environmental problems that lead to autism through the microbiome? We're just touching on the tip of that. We're right there at the surface. We have no idea. And so this is a
Wow.
really great, exciting project that I'm involved in. And I also want to say a lot of people may want to follow up on what I'm saying. So Pilz Bioscience, for your listeners and the followers, is really part of Nova Life Science Corporation. So Pilz was recently acquired by Nova, a public Canadian company. So we're really we're a division of Nova Life Science Corp. And the stock symbol in Canada is NOVA. The stock symbol in the US is LIBFF, Frank,
Okay.
LI Bravo, Frank. But all of this is online. We back it up scientifically. Any questions you have, I don't know, maybe it was Plato who said, or maybe it was Socrates who said, there are no stupid questions, only stupid answers.
Yeah, I love when you say that,
So,
Dr. Hausman.
I back up everything scientifically. Challenge me. You have listeners or readers who want to challenge the stuff we're doing. We'll back it up. We're not afraid.
That's awesome. That's awesome. Evidence-based journalism, evidence-based content. That's definitely the crux and the foundation of what I'm trying to do here with this podcast and the content I create, and bringing experts like yourself here into the mix to talk about the exciting work that you're doing. And so, yeah, of course, like, you guys heard Dr. Hausman say, all the listeners, you're all welcome to reach out. Where to reach Dr. Hausman. You can reach us at info@microdose.buzz. Please ask us questions. Please challenge these very interesting and deep rabbit holes that we're going down. And that's, how science works, and how we have stimulating and intellectual discussion and learn from each other. So that's exciting. I was going to actually ask you about Nova Mentis and Nova Life Science. So it's really exciting to know that, about the takeover and that they're public, and a lot of really big players are going public in this space right now. So I'd love to know a little bit more about your team. I know that you work with a lot of other really skilled scientists like yourself, and what the day-to-day kind of looks like for you guys, working on this
We're
at
growing.
the cutting edge.
We're growing every day. Some of it is still a little confidential. What we've made an announcement of is, I mentioned Dr. Viviana Treza, who was a professor in the Department of Science at Trey University in Rome. She's one of the world experts on autism and the autistic VPA model. So she's now a consultant to the company. We have Dr. Julia Peridiri, who's a neuroscientist from Oregon Health Sciences University and UCLA School of Medicine. She's done work in the past on autism. We now have a consultant, Dr. Kyle Ambert, who's a foremost artificial intelligence expert. His expertise is in machine learning and deep learning. Let me give you an example what that is. If you have, say, 20 sheets of paper in parallel floating in the air, and there's data on each sheet, let's say 20 different data points, 20 times 20, think of the number of variables there, and then you want to look at data point number 10 and see how it changes relative to data point number 111.
Right.
That's called, that's Google Analytics and machine learning. He's an expert on that. So when we have these markers in the microbiome, 50, 100 markers, have cytokines, 50, 100 cytokines, behavioral changes, he can set up the artificial intelligence to analyze all this for us. And he is our consultant. And he got a promotion now. He is a director at Nike Corporation. So we're very proud of our relationship with him. So this is what has been changing on a regular basis. We're also starting to manufacture these derivatives, and you'll hear more about it. We want to be the first company in the world to have an entourage formulation of derivatives from mushrooms, psychedelic derivatives. All of that is ongoing. And you'll hear about it in a regular basis over the next 3 to 4 weeks.
That's really interesting. First of all, congratulations. You have a really dynamic and an all-star team of scientists that you're working with, and experts. And I have no doubt that you guys are going to continue doing really amazing, groundbreaking research. I think it's really cool that you talk about the entourage effect, but then you talk about the derivatives, and in my mind, that's a really interesting combination, because I guess the way I've thought about psychedelic derivatives, it's like a very single molecule synthetic sort of approach, versus when I think of entourage effect, of course, cannabis or all the different alkaloids in mushrooms. Where do those two worlds meet? How does the entourage effect and synthetic derivatives, how do they come together?
You got to start someplace, right? I mean, in nature, it's complex. So in order to prove the entourage effect to the some of the rules of the game are promoted by politics and by government. The rules of the game in the psychedelic industry right now are the regulatory authorities. They want to show an effect of a 95% or pure molecule. So you can't do that in nature right now. You have to do it synthetically. Correct. So my approach to the entourage effect is to play by the rules of the game. Create 3 to 4 compounds that are 95 to 99% pure, and show that the whole is more effective than the parts. Then go to the next level within the food space, the kingdom, and then extract out the molecules I'm looking at and show it again. So another company we're dealing with confidentially is developing extraction technology. Then the next level, I'm giving you my whole chess game, so is going to be delivery technology. No one's thinking about that. How do you deliver this?
Right.
Do you inject it? Do you eat it? Do you use it intranasally? Do you go through, you can do a suppository through the rectum, maybe an autistic child who doesn't want to swallow pills. That's another part of the ball game.
Yeah,
You have to know what
absolutely. Wow.
and
So,
how do we deliver psilocybin?
right.
Right now, it's just oral. Is
Well,
that
it's a
the problem?
prodrug, so it kind of needs to be, that kind of needs to go through the liver to turn into psilocin and be active, correct?
We're going to find that out. You know what happened? What's
Nice.
the receptors in psilocybin? As I said, you know, serotonin 95% is produced in the periphery. Periphery
Right.
is the small intestine, the liver, the pancreas, the stomach, a lot of other organs than the brain. This is where we have to look, and it's exciting. And that's what we're doing. In Nova, we are combining, if you look at our chess game and the people working for us, and we've outsourced. Think of who we've outsourced. We don't have to have everybody internally. We
Mm-hmm.
outsourced one of the largest cytokine flow cytometry companies in the world, Thermo Fisher. We've outsourced
Yeah.
to a company on microbiome genetic analysis. We're outsourcing and we're insourcing by hiring the right people. And I'm not comparing myself to other companies. The proof is in the pudding. We'll see what
Well,
happens in 2021.
well, I'm glad you said that actually, because I was going to ask you to do just that. What makes the team at Nova Mentis and unique and different from the rest. And I think you've answered that really well. But I'd love to give you the floor to comment on it a little bit more. Because there are a lot of people in the space, and I have to say I think this is my 12th episode of the Sci-Fi podcast now. I've had the pleasure to speak with some really dynamic and amazing people. You guys are definitely taking a very unique approach, and I think that's amazing. It's refreshing and it's exciting indeed.
And stay tuned, as I said. It's going to be an exciting 2021. And by the way, the merger, uh, the acquisition by Nova of Pilz, we were in autism. What was interesting is that Nova has shown an interest in psychedelics in obesity and diabetes and a liver disease called NASH, non-alcoholic hepatotoxicity. The information that we're developing in autism in the model with Dr. Trazer has broad application outside of autism. Could be in obesity, diabetes, type 1, type 2 diabetes, liver disease. We're just beginning our research. And so everything in life happens for a reason. Um, Maybe Nova had a greater perception of the future by acquiring us. I don't think they knew exactly what we had till we're now in, so to speak, working together. Um,
Sure.
this could be an unusual relationship now.
Yeah, we're on the cusp of such an enormous, the potential for having such an enormously valuable new toolkit of drugs and therapies and treatments for such a wide range of conditions. And, a lot of people, and in myself as well, when I write about psychedelic medicine and what it's going to treat, I often just say neuropsychiatric conditions and I move on. And here we are talking about diabetes and autism and IBS and so many other applications of psychedelic medicine. And not only the ability to treat them, but through trying to discover these treatments, we're learning so much more about the pathology and ourselves and what makes us human. And for example, that was so fascinating what you talked to me about C-section versus a vaginal birth and how the microbiome is related to that, and so there's so many intersections and so many possibilities to come up with revolutionary and radically effective new treatments, and you guys are really on the cusp of all of it. So what a place to be and what a time to be alive, huh?
Yeah, absolutely. In the pharmaceutical industry, I'm excited about the future for our company. I'm not predicting what's going to happen, but our technology could be applicable to some of the biggest pharmaceutical companies. And then you're seeing
Absolutely.
that in your own space, all these acquisitions. It's
Hmm.
funny, when I see acquisitions, I look and see, why is a company acquiring another company? Is it that the original company doesn't have the technology that the second company has?
Yeah.
Is there a lot of fluff? There's not enough meat on the bones? Do we have to start looking for other technology? When we start a company, we have to rev up our engines by acquiring other technology. It's funny, I've just watched a movie last night, Ford versus Ferrari.
Oh yeah, how was it?
Oh, it was wonderful. My wife had told me to watch it. I love NASCAR, and I love watching the car races. And
Yeah.
Ford wants to catch up to Ferrari, so they go to try to acquire Ferrari, and they get turned down. So the
Right.
acquisition fails. Then they say, well, maybe we have to win Le Mans, and they go ahead and get 1, 2, 3 winning in Le Mans, and that's how they showed who they were. Well, maybe that analogy applies to our industry as well.
Oh, man, that's really awesome. Dr. Hausman, thank you so much. As we're coming to a close in our discussion here, I like to always give my guests the last word, a thought, a message that you want to share with our audience about yourself, what you're up to, the future, or anything you want to. The floor is yours.
Thank you. Well, I'm getting old. I had a good life. I had a huge practice in pediatric kidney surgery at UCLA and Cedars. Uh, and you go into this world you come into the world with your name, and you leave with your name. You don't leave with anything else.
Sure.
And if I could leave a legacy for healthcare with my name in autism spectrum disorder, that would be one of my greatest accomplishments.
I think that's a really noble pursuit. My best friend and he's my roommate freshman year of college, and we're still very close. Dominic Vita, he's about to get his PhD, he's studying Fragile X syndrome, at Vanderbilt, and so it's been really inspirational just to see his dedication. And PhD students, they work so hard, but it's been really great to see his passion for autism. And of course, like you said, it's kind of like so many other things today. It's usually just 1 degree of separation. We all know somebody, if not immediately in our family, that's affected by this. And it would be so amazing to get to that next breakthrough in treatments, and therapies for people with autism and beyond, like you talked about, diabetes, IBS, and of course the neuropsychiatric indications that we're exploring. And so, yeah, this has been such an amazing conversation. Of course, I look forward to the next one. I know we'll have so many more. And I'm so thankful for your time. Thank you for coming on the show and, connecting with me, so we could have this great chat.
It just reminded me of something. I mean, I just thought of something. Yes, how I could.
Sure.
I'm willing to help any way I can. Knowledge, communication, and knowledge is beautiful for people. People have to become their own advocates. You have to be your own advocate. You have doctors out there, but they don't call you to see how you are. They only see you when you're sick. And that's
Sure.
an important role. I'm a doctor. But you have
Mhm.
to advocate all the time for yourself. And we just hired a lady who in our public relations department. Her first name is Tara, Tara, and she's available. So if people want to write to you, it would take the pressure off me. But through Tara, we could have an ongoing blog. If people have specific questions on diabetes, heart disease, how it applies to tryptamine derivatives and this could be fun. It's a give and take
Yeah.
knowledge, and I'd love to help if I can.
Well, that's awesome, and absolutely, we've only scratched the surface. So, like you said, I encourage everyone listening to reach out with questions, comments that you guys have, so we can explore these exciting topics further. And you can reach out to Dr. Hausman, and you're obviously welcome to reach out to me at info@microdose.buzz. You can find this podcast on iTunes and Stitcher. And yeah, thank you so much, Dr. Hausman. This has been another great episode of the PsyFi podcast, where we connect with leading industry experts, researchers, and clinicians to unravel the mystery that is psychedelic science. I was here with Dr. Marvin Hausman today, and we'll see you all next time. Thank you so much. Thanks,
Thank you.
Dr. Hausman.
I'm proud to be part of your help, your organization, and part of what you're trying to promote to the world in health. Thank you very much.
Thank you so much. That's our pleasure. Thank you so much, Dr. Hausman.
Thanks for joining the Psy-Fi series, brought to you by Microdose and the Conscious Fund. Visit our website at www.microdose.buzz.